to introduce one of our global editors, Dr. Nicola Conran, and she will introduce today's speakers for you.
Okay, so thank you very much, Jessica. As Jessica said, I'm the global editor for Latin America for experimental biology and medicine, and I work as a researcher at the University of Campinas, São Paulo, Brazil. So it's my great pleasure to introduce our two speakers today for the third and final SEBM webinar, which is on pathophysiological mechanisms in sickle cell disease. Our first speaker will be Guillaume Letry. Dr. Letry received a PhD in developmental genetics from the University of Zurich in Switzerland in 2005. And he's been a professor at the Université de Montréal and the Montreal Heart Institute since 2009. And so he does some very important work in his lab where he uses human genetics and functional genomics to study the sickle cell disease. Then our second speaker will be John Boucher. Dr. Boucher earned his PhD from Wake Forest University and is now Associate Professor of Medicine at the Medical School of the University of Minnesota, and he's been there since 1996. Dr. Boucher's work really has been very key to understanding the role of heme in the pathobiology of sickle cell anemia, and he's shown how hemoglobin, heme, and iron exert toxicity on the endothelium, and also how heme-mediated inflammation can trigger vaso-occlusion in sickle cell anemia. So Dr. Letry will be talking today about the use of Mendelian randomization to study clinical heterogeneity in sickle cell disease. And then we'll have Dr. Boucher talking about the role of heme and toll-like receptor 4, MD2, in sickle cell disease pathophysiology. So I'd like to thank both speakers today for taking the time to give us presentations today. And we'll have a question session for both speakers at the end of the two talks, if you'd like to send your questions in the chat box. So I'll hand over to Dr. Letry if you'd like to share your screen, and thank you very much.
Great, thank you, Nicola. Thank you, members of the Society, for inviting me. Great pleasure to present today, although it's virtual. We're getting used to it, but it's nice to be with all of you. So as Nicola mentioned, I'm going to be describing some of the work we've been doing in the last 10, 15 years using genetics to study this simple Mendelian disorder called sickle cell disease. And I emphasize that although it's been described as a simple disease because it's due to a mutation in a single gene, it is all but simple. And this is I mentioned sickle cell disease is fundamentally a disease of the blood, and yet it is characterized by a multi-organ process with defect in multiple organs occurring throughout lifetime. And although we know that sickle cell patients can suffer any of these, we have a really bad we have really bad tools to be able to predict which patient will suffer from which complication and when. And as you can imagine, this would be extremely useful in order to stratify patients that are more likely to have severe disease. So if we summarize what clinicians have in terms of helping or taking care of sickle cell patients, we can think about prediction, and in these relate to biomarkers. And we do have a few, and these include complete blood count, hemoglobin level, fetal hemoglobin level, brain imaging. So there are some biomarkers which are extremely useful, but far from perfect. And that's for prediction. Now, for prevention and treatment, we also have a few therapies, and we'll discuss some of them today. Blood transfusion, which has been used for a long time. Hydroxyurea also is being used for a long time, and then gene therapy. And although these treatment options are good options, they're not perfect. They can have side effects, and in terms of gene therapy, they're not available to most of the sickle cell disease population in the world. So clearly, there's a need to develop other tools to take care and manage sickle cell disease patients. And the way I look at this problem, I think there's three different strategies to improve sickle cell disease patient care in the long run. I do believe we need more observational studies, mostly in Africa, but I would argue also in developed countries like Canada and the US to better understand the epidemiology and the natural history of the disease. We certainly need more clinical trials in this patient population to test more drugs, to have better, more options in terms of therapy. And what I'm going to emphasize today is we need high quality and collaborative biomedical research with sickle cell disease patient as a collaboration with sickle cell disease patient, to be able to generate more hypotheses that we will then study in large population, which will hopefully bring ideas for new clinical trials. And the focus of my lab and the focus of the talk today will be how we can use omics to develop this research program that takes advantage of newest technology to tackle sickle cell disease heterogeneity. I'm a human geneticist. I like omic research, and there's many reasons why I like it. One of the major strengths of omic is that it generates hypothesis. So we can do a genetic experiment, a transcriptomic experiment, and we will generate either interesting data that will generate hypothesis. It's really, really powerful at identifying what I call features. And these features can be genetic variation, mutation, gene expression signature, metabolite, proteins, essentially anything that you can measure in patient that will correlate or associate with clinically relevant outcomes, such as stroke or painful crisis. But we have to bear in mind that when we do such omic experiment, largely what we're finding are association, and as you all know, association does not imply causality. And that's a major issue, not so much in terms of biomarkers. I mean, there's many biomarkers which are not causally impacting human disease, yet they are extremely useful in medicine. But if you're thinking about drug target, obviously, if a biomarker is not causal for disease, it becomes de facto a bad drug target. And what I want to try to convince you today is that we can use human genetics and some of its recent methods to solve the question of association versus causality in omics data analysis. Controlled trial or randomized clinical trials. So in such experiment, as you all know, we recruit participants. Some of them will be exposed to, say, a drug. Others will be exposed to the placebo. We assume that the confounders are the same between two groups. And as such, if we observe differences after the trials, we conclude that it has to do with the drug and not any of the other possible confounders. In the Mendelian randomization experiment, the drug is replaced by a genetic variant. So again, you have participant, in our case, sickle cell disease patient, which will have inherited at birth randomly alleles that will increase a biomarker or increase a molecule that we are interested in, and others will have the other allele. This is a random process due to the segregation of allele during conception. Again, we assume that confounders are the same between the two groups, and again, as such, if we observe difference in terms of the clinical outcome, we conclude that it has to do with the allele, which is associated with the molecule or metabolite or protein that we are interested in studying. Just to clarify or crystallize this concept, I'll give you one example where Mendelian randomization has been extremely useful to confirm the causal role of LDL cholesterol in coronary artery disease risk. So in randomized controlled trials, we can divide individuals at risk of heart attack between those that take statin and those that take placebo. And sure enough, statin will lead to lower LDL cholesterol and lower risk of reduced coronary artery disease. We identify these individuals at lower LDL cholesterol, and then another group of individuals that do not have these alleles, so they have an unchanged or average level of LDL cholesterol. And when we do this experiment, we will observe that individuals that have genotype or alleles that lower LDL cholesterol are at lower risk of, again, cardiovascular disease. So it's essentially using genetics as the intervention, as the drug that we're testing. So now let's try to apply this more concretely in the sickle cell disease. Oh, and just before going into the results, just bear in mind that Mendelian randomization is a relatively straightforward statistical method, but it's a variant, which is the instrument that would have an impact on the exposure. In my previous example, the exposure was LDL cholesterol, and then which will have an impact on outcome. Again, with the previous example, was coronary artery disease. Some of the assumptions are the following. First, the genetic variant has to have an effect on the exposure. The genetic variant cannot have an effect on the confounders, and the genetic variant has an effect on the outcome only through the exposure. So under these three assumptions, which are sometimes difficult to validate, Mendelian randomization is a valid statistical strategy to confirm whether or not an exposure, a molecule, a metabolite, a signature is causally related to the outcome. Okay, so now let's try to apply Mendelian randomization to our sickle cell disease complication. And we'll start with something very easy. It's already well known that bilirubin level, plasma or serum bilirubin level, is associated with gallstone and gallbladder disease. This is true not only in sickle cell disease patient, it's true in anybody, but it is more frequent in sickle cell disease patients. So let's say we wanted to confirm that bilirubin level, high bilirubin level, cause gallstone disease, and it's not simply a biomarker of this complication. So what we would do is try to identify a genetic variant that are associated with bilirubin and test their association against bilirubin as well as with gallstone disease. So we can do that in sickle cell participant here. We have a cohort of African-American participants from the large CSSCD cohort, so 1,278 participants. We have one genetic variant at the UGT1A1. It's a gene that's really well known for its role in bile production. And what we see is that this variant is strongly associated with bilirubin level, a p-value of 9.7 times 10 minus 21 in this cohort, and is also strongly associated with gallbladder disease. So it represents a good instrument for these experiments. Now let's do the Mendelian randomization. So we find variant associated with bilirubin. So we found in total 10 SNPs, so 10 genetic variants, which we validated for the different MR assumption and did the calculation. Again, I didn't list how the math work here, but bear in mind that Mendelian randomization, the framework is relatively simple. And when we did all of this, applying it to our cohort, what we observe is the following. When genetically you increase bilirubin by 2.3 milligram per DL, that's one standard deviation, you increase sixfold the risk of gallbladder disease in sickle cell disease participants, thus validating what was already known, that is the link, the causal link between bilirubin and gallstone, but also validating the approach in our patient population. All right, so that was easy. Let's do something a bit more interesting. Let's try to quantify the impact of the known modifier fetal hemoglobin on sickle cell disease complications. So it's been known for a long time that fetal hemoglobin is a strong modifier of severity, and patients with a high level of such hemoglobin have milder disease. So we did the same thing. We identified genetic variants associated in 3 genes that are associated with fetal hemoglobin, calculated the MR statistics, and what we found is that increasing by 4%, so genetically increased 4% fetal hemoglobin, reduce the risk of acute chest syndrome, a complication related to the lung, reduce the risk of stroke, but in this design had no effect on painful crisis, which was a bit surprising. I would cautious overinterpretation of this result. I don't think it has to do with the fact that fetal hemoglobin doesn't have an impact on painful crisis. I think it relates more to the clinical definition of painful crisis that we're using for these analyses. So it's working. So we can quantify fetal hemoglobin levels and its impact on sickle cell complication. Fine. So now we have a framework that works. What can we do next? Well, I mentioned earlier that part of my lab is interested in finding biomarkers and drug target for sickle cell disease. And we, a few years ago, decided to take a metabolomic approach. So we collected plasma from 705 sickle cell disease participants, and we measure a very, very large number, thousands of metabolites. But for today's talk, I'm going to focus on 129 known metabolites that we measure in these 705 participants. And then using this data, we ask first, are these metabolites correlated with complication? And then can we use Mendelian randomization to validate their causal effect on these complications? Before presenting all of the results, I'm going to show you the result for one metabolite that we were particularly excited to look at, and that's L-glutamine. The reason why we were excited is because in 2017, the FDA has approved L-glutamine as a treatment for sickle cell disease. It is a metabolite. It's one of the most abundant amino acid, and it's involved in changing the NAD redox ratio in red blood cells. And as I mentioned, in a recent clinical trials published in 2018, it was shown that L-glutamine reduced painful crisis over a 48-week period. So we were interested in testing, confirming the clinical trial using Mendelian normalization in our experiment. First, we tested the association between the L-glutamine that we measure and retrospective phenotype, so event that occurred prior to the beginning of our experiment. And there, slightly disappointed, we were slightly disappointed. We saw no correlation or association, if you want. So L-glutamine that were measured on a given time on day 0 of our experiment were not associated with clinical event that occurred prior to the metabolomic experiment. And there's many reasons why this could be the case. So then we wonder, could we use Mendelian normalization and variants that control L-glutamine levels, so variants that were there at birth are there since the beginning, and see whether these variants, as instrument of L-glutamine variation, associate in the Mendelian normalization framework with painful crisis. So this is the experiment that we did, and this slide is complicated. We identify, you see here at the bottom, 51 genetic variants that are associated with L-glutamine levels. And we derived 2 statistical models, and I will focus your attention on model number 2. The model number 2 is the one that respects the assumption of Mendelian normalization. And when we did this, what we found for painful crisis is that the 1 standard deviation increase in L-glutamine level in the large CSSCD was associated with a 32% reduction in painful crisis risk, and this was significant. We attempted to replicate this result in a smaller cohort, about half the size of this. And although we noted a similar effect in terms of the direction, again, reduction, the result was not significant, which could be implying that the sample size was not sufficient to capture this effect. As for the clinical trial, we observed no other significant association in our MR analysis between L-glutamine and other complications such as gallbladder disease, retinopathy, leg ulcer, priapism, and necrosis. So some form of validation that L-glutamine is indeed causally implicated in painful crisis in sickle cell patient. This was consistent with conclusion from the clinical trial. So that was very encouraging. We then turned our attention to all the other metabolites that we had measured, and we tested them against the main complication that were available in the clinical chart from our sickle cell cohort. And we identified 2 metabolites associated with painful crisis, 2 metabolites associated with gallbladder disease, and 62 metabolites associated with EGFR, which is a quantitative measure of kidney function. We use the same strategy. So we first identified genetic variant associated with the level of these metabolites, and again applied the Mendelian normalization framework to test their causal effect. And for all of these metabolites except 1, we saw no causal effect. The only metabolite for which we observe a causal effect in this discovery cohort was a metabolite called 3-ureiodopropionate. There's very little known about this metabolite. What you observe here is on the x-axis, the effect of the SNP on the metabolite itself versus the effect of the SNP on EGFR. And because the slope is different than 0, it's suggesting that there's a causal effect between the two. This was highly significant in the CSSCD in one of the large cohort, but non-significant in the smaller cohort. So this is clearly unreplicated and will need further validation. Finally, one last thing that we try to do is to take all our data, all the metabolite that we had measured, and determine whether we could build predictive model of mortality in our sickle cell cohort. So we had access to data with close to 600 patients, including 54 individuals that passed away during the follow-up period. And we tested all our 129 known metabolites against this phenotype. And we noted that many of those were associated with mortality. In particular, we noted that quinolonic acid was significantly associated with increased mortality risk, even when we corrected for all the different metabolites and tests that we had done. Interestingly, Another group has previously reported that quinolinic acid is associated with rapid renal function, so sickle cell disease nephropathy. And sure enough, when we adjusted our association analysis with EGFR, we lost the association, again suggesting that many of these metabolites that are associated with mortality do so by reflecting the activity of the kidney, so capturing, if you want, some aspect of sickle cell disease nephropathy. So in conclusion, Mendelian randomization is a powerful method to complement omics-based research to identify causal molecule and prioritize drug target. It has some limitation, sample size. We need large studies to do MR, and this is limiting for sickle cell disease. And another limitation is that we need more genetic data in individuals of non-European ancestry. This is certainly one of the big limitations we have in human genetics right now, and hopefully in the near future, we will have more data in individuals of non-European ancestry so that we can derive better instrument to implement MR in sickle cell cohort. We provide some evidence of causality between L-glutamine and painful crisis. The reason why I'm hesitant to say unequivocal, very, very clear evidence is simply because one of the cohort didn't replicate the association. And then we identify 66 metabolites that are associated with sickle cell disease phenotype. So these could potentially be very great new biomarker of complication, and we are awaiting validation for some of them in other cohorts. I want to acknowledge Jan Ilbudo, who's a very talented graduate student in my lab who performed most of the analysis that I described, our collaborators in the US, as well as in France, as well as the funding agencies. And with this, I'll stop. I guess I'll stop sharing, and then we can listen to John's talk.
Okay, thank you so much.
Okay, thank you. That was a great talk. Maybe I can shed some light on an area of the biology that may be contributing to some people having more severe disease and some people having less severe disease. I've been asked to talk about the role of heme and TLR4-MD2 in sickle cell disease pathophysiology. Our lab receives research funding from CSL Behring and Mighty-Bridge Astellas. Hemoglobin S polymerizes under deoxygenated conditions, such as when the red cells traverse the venous system. And this can lead to extravascular hemolysis and intravascular hemolysis through the change in shape of the red cell. I'm going to focus today on intravascular hemolysis in today's talk. So during intravascular hemolysis, hemoglobin gets released into the vascular space, and then the iron is released and stored safely in ferritin. But in sickle cell disease, haptoglobin levels are depleted because of the constant rate of hemolysis that's ongoing. And this free hemoglobin can be oxidized by metabolites such as nitric oxide to methemoglobin, which is iron 3+. And methemoglobin is unstable and can readily release its free heme or hemin. And this hemin, again, under normal circumstances, binds very, very tightly to a plasma protein called hemopexin, which carries it primarily to hepatocytes, to LRP1 receptors. Again, it gets endocytosed, the heme gets degraded in the vasculature. The heme ring can be opened up by exposure to cells and other cells. That can release, once that ring pops open, it can release free iron and catalyze more reactive oxygen species and cause inflammation in the vessel wall. Just to give you a sense of how much heme is actually being released and degraded in a sickle mouse, heme degradation by heme oxygenase produces 1 mole of carbon monoxide, or CO, for each mole of heme that gets degraded. This CO is primarily carried by hemoglobin to the lungs where it's expired. And so you can measure the expired CO in patients and in mice. In a Towns SS mouse, which has a RBC half-life of 2.5 days, we the mice expire 6 times more CO per gram body weight than a hemoglobin AA mouse, indicating that hemoglobin heme turnover is 6 times greater in SS mice compared to AA mice. Let me just, do I have is my video off?
No, you're on.
Okay. All right. So as a result, as I mentioned, plasma haptoglobin and hemopexin become depleted, and this free heme can be taken up by the vessel wall through non-receptor pathways. So we've been interested in this question: does this free heme promote inflammation? And if it does, how does it do so? In 1997, Frank Wagner and Nader Abraham's lab published a paper that showed that heme induces the expression of adhesion molecules on vascular endothelial cells. Carl Nath at the Mayo Clinic published a paper showing that heme induces chronic renal inflammation, and they could suppress that inflammation by inducing heme oxygenase 1. So we were interested in how does heme produce inflammation, and is the innate immune system involved? A landmark paper was published in 2007 by Marcelo Bozo's lab in Brazil. And basically what they showed was that heme can induce mouse and human macrophages to secrete TNF alpha. And they went on to show that this was dependent on TLR4 signaling. When they took macrophages from TLR4 knockout mice, both heme and LPS, which is the canonical ligand for TLR4, both lost their ability to produce TNF alpha. Similarly, if they used MYD88 knockout mice, MYD88 is a signaling molecule that attaches to the intracellular domain of TLR4, is required for signaling. If they deleted, they had macrophages from MYD88 mice, again, heme and LPS lost their ability to produce TNF alpha. And same with a CD14 or another known required cofactor, although it wasn't quite completely knocked down in the LPS mice, knockout mice, macrophages, excuse me. So the signaling here on the left-hand side of this diagram, LPS and heme induce dimerization of MD2 TLR4 complexes. This brings together the cytoplasmic TER domain, which recruits adapter molecules like NYD88, which then through a signaling cascade activates NF-kappa B, transcription factor that activates the transcription of pro-inflammatory cytokines and adhesion molecules. There's also a non-MYD88 independent pathway where that leads to a late phase NF-kappa B response, as well as interferon gamma gene products. This is a model that was Samit Ghosh and his colleagues at the University of Pittsburgh showed that free heme could induce death in SS, but not AA and SS mice, shown here in this diagram. They also showed that if they gave the heme with hemopexin, it prevented the cell death, or if they added the TLR4 signaling inhibitor TAC242, that could improve the survival. They also showed that the heme induced lung injury in these SS mice, congestion, edema, alveolar wall thickening, and hemorrhage. And that if they gave TAC242, it prevented this lung injury in response to heme. Or if they had a TLR4 knockout in the lung tissue, also prevented the injury. So we wondered in our lab if heme also activated endothelial cells. And what we showed is that heme triggers really rapid viable body P-selectin and von Willebrand factor expression on the surface of UVEC in vitro. So P-selectin in green and von Willebrand in red. This is cell surface expression. You can see the positive control histamine induces viable body release of these proteins within 2 minutes. Heme, within 5 minutes, we saw a lot of expression of P-selectin and von Willebrand factor. Similarly, this also happens in vivo if you infuse hemin. This is a much lower non-lethal dose of hemin. In every tissue we looked at, this is just the lungs here, in C57 or in a sickle mouse, the hemin induced the expression of B-selectin and von Willebrand factor on the vessel wall within minutes. We showed that then TAC242 could inhibit the viable Pilates-body activation by hemin, shown here on the panel on the right. We also showed that heme could activate NF-kappa B, the pro-inflammatory transcription factor I just mentioned, in endothelial cells via TLR4 signaling. So in panel A is UVEC that have been incubated with LPS, TNF-alpha, or heme for 1, 4, or 8 hours. You can see all of those treatments induced NF-kappa B phospho-P65, which is a marker of activation. However, if we added TAC242 to that, it blocked the activation. Similarly, in pulmonary vein endothelial cells, in wild-type mice, heme, LPS, and TNF induced NF-kappa B activation. The heme could be blocked with TAC242, or if we use pulmonary vein endothelial cells from TLR4 knockout mice, heme and LPS lost their ability to activate NF-kappa B, but TNF-alpha did not. So we then wondered, okay, since it's affecting the viable Pilates bodies and P-selectin, which has been implicated in vasoocclusion, we wondered if heme would also induce vasoocclusion or microvascular stasis. So we used a dorsal skin fold chamber model to measure microvascular stasis in sickle mice in response to heme and hemoglobin. Here, when you infuse saline, you see very little microvascular stasis 1, 2, 3, and 4 hours after the infusion. But if we infuse normal hemoglobin A, we got up to about 40% microvascular stasis in the model. Similarly, if we infused hemin, we also got a similar amount of stasis. Or if we infused water, which causes lysis of the red stasis. We then wondered if the heme was coming off of the hemoglobin. So in this experiment, when we infused methemoglobin, which as I mentioned is unstable and releases its heme, we see stasis in the model. However, when we infuse cyanomethemoglobin, which stabilizes heme, and so the heme does not come off of hemoglobin, we did not see induction of stasis. Also, when we infused hemoglobin, again, we saw stasis, but if we added haptoglobin, which binds hemoglobin and prevents the release of heme, we saw very little stasis. Similarly, and this was a very interesting experiment, hemopexin also blocked hemoglobin, suggesting again that the heme is coming off of the hemoglobin after it's infused. So does TLR4 mediate microvascular stasis? We bred a TLR4 knockout mouse onto the Towns SS and AA mouse model. This just shows you the PCR gels. Lane 1 is a C57 knockout mouse. You can see here the knockout mice have this 140 base pair band. Towns 4, we also had to make sure we got rid of the murine globins, and they're only expressing the human alpha and the beta globin. And again, you can see the human alpha globins here. The beta A and beta S, beta A, beta S bands, human in the AASS and the AA and SS knockout. And here's the C57 murine. So we had a very nice model here, which we then back crossed for 10 generations to homogenize it with our colony. We then looked at microvascular stasis in response to hemin, LPS, or hypoxia and reoxygenation. And the SS knockout mice, we were able to make chimeras where knockout recipients were A mice with TLR4 minus minus plus plus minus minus. So at the end of the day, when we measured microvascular stasis, we saw that if we knocked out TLR4 in the circulating blood cells, but not in the vasculature, we got microvascular stasis. Similarly, if we knocked out in the blood cells and in the endothelium, we were able to inhibit stasis. And down here, we found that if we had TLR4 in the blood cells and in the vessel wall, we also got stasis as we expected. But when we knocked out TLR4 in the not in the hematopoietic cells, but only in the vessel wall, we were able to inhibit stasis. So this confirmed that it was the endothelial TLR4 that was important for microvascular stasis. So in our model, hemoglobin S releases heme. This can be blocked by haptoglobin, or the heme can be bound by hemopexin. But if you have free heme, it can bind to the TLR4-MD2 complex, leading to signaling activation of NF-kappa B and Weibel-Palade body degranulation, which then promotes the microvascular stasis or vaso-occlusion. So how does heme initiate TLR4 signaling? A lot is known about LPS TLR4 signaling. The binding site, LPS binds to MD2 to initiate that signaling. You can see the binding site here on MD2. It's been very well worked out. So we wondered, does heme have a binding site on MD2 similar to LPS? We did first we wanted to see, does heme actually bind to MD2? And so in this experiment, we used a strep avidin agarose pull down assay with biotin heme. So in this reaction, we use recombinant MD2 from transfected CHO cells. You can see here the recombinant MD2. It has a flag tag. So when we do the Western blot on this image, we're actually doing anti-flag. Okay, so here you can see there's plenty of recombinant MD2 in the starting CHO media. We then incubate that CHO media with biotin heme, and then either agarose or streptavidin agarose and pull it down. So if biotin is binding to MD2, then it should pull down the MD2 with the streptavidin agarose, but not the agarose. And that's what we found. You can see if without the streptavidin on the agarose, we didn't pull down any biotin. Okay, is MD2 required for TLR4 activation of NF-kappa B by heme? So we transfected HEC293 cells with plasmids encoding wild-type MD2, TLR4, CD14, and we used an NF-kappa B luciferase reporter. So this is a luciferase gene with an NF-kappa B binding site. So whenever NF-kappa B is activated in these cells, we get a signal from the luciferase. These transfected cells were stimulated with heme or LPS or both for 4 hours, and then we measured the NF-kappa B luciferase activity. You can see in control cells, we got very little luciferase activity. However, if the green bars, if these cells were transfected with TLR4, MD2, CD14, and of course the luciferase reporter, you can see we got induction of luciferase activity with heme, with LPS, or the combination of LPS plus heme gave us even more. But importantly, if we left out plasmids for CD14, for MD2, or for TLR4, the purple, blue, and orange bars, we got no induction of luciferase activity, indicating that all three of these proteins are necessary for heme activation of TLR4 signaling. Now, this was already known for LPS, but MD2, that was a new observation that had not been shown. So we then wondered where the heme was binding on MD2. So we did an in silico analysis with our colleague at the University of South Carolina, Dr. Hu. He has a heme bind program, which is actually available online. And this program uses the structure of the protein, in this case MD2, and the amino acid sequence of the MD2, and it identified 2 clusters that were potential heme binding amino acids on MD2. And I've shown those 2 clusters in magenta and green here. So then we mutated these amino acids to alanine to find out what the effect would be on the NF-kappa B luciferase assay that we just did. So wild type MD2 was set at 100% here of luciferase activity, is shown by the dashed black line. On the left here is heme stimulation, on the right is LPS stimulation. Focus over here on the heme stimulation. Again, here's wild type MD2, along with, of course, TLR4, CD14. And you can see when we mutated the tryptophan at position 23, we lost activity. When we mutated the serine at position 33, we did not. When we mutated the tyrosine at position 34, we got a strong depletion of activity. However, when we mutated the amino acids in that green cluster, you can see that we actually got some stimulation at position 36, but no effect at 37 and 44. When we looked at LPS stimulation, mutation of the magenta cluster had very little effect on TLR4 activity, and also mutations in the green cluster had little effect. So it appeared that these two mutations, these two amino acids, may be where the heme was binding on the MD2, shown here and here. So we then did a heme binding assay, again, streptavidin and agarose and biotin heme. Here you can see wild type MD2 and the mutation at 23A. And here's the triple mutant 23, 33, 34. We got reduced heme binding when we use these mutated amino acids. Over here is the control CHO media that's been transfected, similar amounts of the MD2 in those three examples. And then here's the quantitation of these three bands down here on the bottom. So we then got John Trent at the University of Louisville, a structural biologist, to give us where is this actually, how does this look? And so what it turns out that this heme binding site is actually on the opposite side of MD2 from the known LPS binding site shown here. So we then moved on. Well, okay, MD2 can be bound to TLR4, but it can also be found in circulation in the plasma. And it's elevated in various inflammatory conditions, this soluble MD2. So we wondered if the soluble MD2 is playing any role in endothelial cell activation in SCD. So the first thing we did was we measured soluble MD2 levels in human and mouse SS plasma. Here's a Western blot. And the quantitation of those Western blots showing an increase in soluble MD2 in SS plasma in human and mouse. We also did ELISA on human plasma and showed that there is an increase in MD2. It looks to be somewhat bimodal here. We then put SS and AA plasma on endothelial cells. In this case, this example is human lung microvascular endothelial cells. We also did this in UVEC. And in both cases, SS plasma increased the secretion of IL-8, soluble VCAM1, and IL-6 in the media of the endothelial cells after 18 hours. And this increase, here's IL-8 example, was blocked by adding TAC242 to inhibit TLR4 signaling. So is MD2 involved in this? Well, we then depleted SS plasma of soluble MD2 in approach 1. In approach 2, we added soluble MD2. Again, this is recombinant to AA plasma. We then incubated that with UVEC and looked at IL-8 secretion. So here's the depleted SS plasma. You can see we depleted the SS, the soluble MD2 in that plasma. And that when we incubated the depleted plasma with UVEC, we saw a decrease in the amount of IL-8 produced by that plasma. Similarly, when we added back soluble MD2 to AA plasma, you can see here we added soluble MD2, incubated it with UVEC. We increased the IL-8 secretion, but that could be blocked by TAC242. We then showed again one more time, this is in a different publication that's in press, that soluble MD2 in plasma. Before we showed it with recombinant, this is using this MD2 in plasma, also binds heme, both human and mouse SS plasma. You can see this is a heme agarose as opposed to streptavidin agarose and biotin heme. We used heme agarose and showed that the heme agarose pulls down both human and mouse soluble MD2, but control agarose without the heme does not. We then also did a absorbance spectra, and you could see the Sorey band at 414 increases when we have soluble MD2 plus heme. Here's a positive control, hemoplexin plus heme. Interestingly, that without adding the heme to the blue line here, you can see there's already a little bit of heme that appears to be bound to both hemopexin and MD2 here in this example. We then showed, okay, is the heme involved in this signaling with the heme bound to the MD2? So here's AA plasma, IL-8 secretion again, SS plasma. If we add hemopexin to AA plasma, we got very little decrease. If we added hemopexin to the SS plasma, you can see we got a significant decrease in the amount of IL-8 secretion. Over here, if we added soluble MD2 to AA plasma, shown here, one is AA plasma alone, and this could be inhibited by adding hemopexin. Similarly, if we had SS plasma and added more soluble MD2, we increased the IL-8 production even further, and that was blocked by adding hemopexin. So in summary and conclusion, we've shown heme activates endothelium through TLR4 signaling. It promotes microvascular stasis, and microvascular stasis in response to heme, LPS, and hypoxia reoxygenation is ablated in TLR4 knockout SS mice. We showed then that the loss of endothelial, but not the hematopoietic TLR4 prevents the heme-induced microvascular stasis, required for TLR4 signaling by heme, and that heme binds to MD2 at a site that's independent of LPS. And then soluble MD2 is elevated in human and mouse plasma, SS plasma, and soluble MD2 bound to heme and plasma activates endothelial TLR4 signaling. We speculate that targeting that heme binding site on MD2 may inhibit the heme-mediated vaso-occlusive crisis while preserving immune responses to LPS. I just want to thank my colleagues at the University of Minnesota. Greg Versalati has been my scientific research partner for many years. So is Bob Hebel. Dr. Chen has done all of the stasis. Fuad Abdullah maintains the mouse colony and did all the breeding for the TLR4 knockout and make sure we have the right phenotype and genotype. Ping Zhang is our molecular biologist, and she did all of the soluble MD2 work I just showed you. Joan Beckman did the transplants and the studies with the TLR4 knockout mice. And John Trent at the University of Louisville is the structural biologist that helped us with the MD2 heme binding structure. So with that, I'll pause and hand it over to for questions.
Thank you very much, Dr. Boucher, for an excellent talk too. And I'll hand over to Wenon Zhao, who's going to be coordinating the questions and answers session.
Okay, thank you.
Thank you.
Yeah, I'm Wenon Zhao. I'm a training counselor at SEBM. Okay, so I have a question for Dr. Leiter. So which types of food are rich in L-glutamine, and what are the side effects of glutamine consumption? Yeah, thank you.
So L-glutamine is an amino acid. So any protein will contain L-glutamine. It is one of the most abundant protein in the body, and I don't think that there has been study that have looked at the over intake of L-glutamine, at least as far as I know. So I wouldn't know. What I can tell you is that above and beyond L-glutamine being used in the clinical setting for sickle cell patient, it can also be bought at any store that sell nutrients. So it's widely available.
Yeah, thank you. There is a question for Dr. Belcher. Would the TNF-alpha signaling disruptor be helpful for patient crisis?
Well, I think so. I think you have to think of it in two ways. One, molecules that might prevent TLR4 signaling, and molecules that might resolve a vaso-occlusive crisis. So currently, it's not directly TLR4, but it's hemopexin is getting ready to enter clinical trials. And it'll be used in a treatment modality where it's given to patients when they come in to the emergency room in crisis. The thought being, in preclinical models, at least we have shown that it actually resolves vaso-occlusion much quicker than if you just give a vehicle. TLR4, TAC242 failed clinical trials for sepsis, but one of the issues is always going to be keeping the immune response intact. You want to be careful that you don't give a drug that's going to allow bacterial infections. And so that's why we think targeting the TLR4 heme binding site makes a lot of sense. And we're well aware heme's not the only activator in sickle patients. They go in crisis for lots of reasons besides just hemolysis, but it might be an approach that might work inhibiting TLR4 signaling to prevent a crisis, because there seems to be a lot of things that turn on that pathway that induce stasis in our model.
Thank you. So here is another one for Dr. Leiter. How do you see this omics study shaping precision medicine for sickle cell disease in the future?
So it's a good question that I have thought about quite a bit. What we need is larger studies, because right now the field of sickle cell disease precision medicine, or whatever you want to call it, is really hampered by the fact that our cohort remained very, very small. And there's no reason to that, because there are thousands, tens of thousands of patients out there which would benefit from these studies. Where I think this is going is, as for most other chronic human disease, it's going to a world where we will be able to stratify patients, so identify those that are more likely to suffer from very severe complication, those more likely to benefit from drugs. And that's in the short term. I do believe that this is very reachable. And then in the longer term, we might be able to identify precisely who will benefit from that drug or that gene therapy option. So I think there's a short term, long term, but I cannot see us reaching any of these goals unless we massively invest effort, energy, collaboration in larger sickle cell disease study.
Oh, thank you. There are a couple questions for Dr. Belcher. So the first question is, what about, you have shown that MD2 bands heme. So what about a direct binding of heme to TLR4? Did you exclude this, or it is still possible to have a second binding to TLR4? Thank you.
Yeah, thank you for that question. I think that our experiments where we did not include MD2, in other words, when we lost signal by leaving out MD2, suggests that MD2 is required. But to answer your question directly, no, we didn't.
Okay,
I know that's an indirect answer, but that's the best I can do, because we didn't look at it directly, and so it's certainly possible that heme could bind to TLR4 as well as MD2.
Yeah, thank you. Additional questions. So is there any evidence for the MDA2 heme binding to TLR4 on other cell types in addition to the endothelial cells?
Yes, absolutely. Platelets have it. Leukocytes have it. Lungs, interestingly, pulmonary epithelial cells have TLR4, but they don't have MD2. They're entirely dependent on MD2, soluble MD2 being delivered from plasma exudates. So yeah, many cells have this pathway that could be, and I think platelets are very fascinating. That could well be very important TLR4 signaling in platelets induced by heme and other molecules.
Yeah, thank you. I'm also interested in this cell type specific also have different transcriptional regulation by heme, or they are the similar across different cell types.
No, I think vascular endothelial beds probably vary very much in other cell types besides endothelium. So no, I'd say there's a lot of variety. You know, I think we're seeing that with COVID. I think we're seeing, you know, the vascular beds seem to be affected differentially. And I think that's differences in the innate immune system. Dr. Rumini is on the line here, and she, I'm sure, could talk very important in sickle disease. So I think it's going to be, you know, I think the lungs is obviously and the kidneys are obviously very important targets in sickle disease, but we see disease, you know, pathology that manifests in many different ways. So I think it's probably a heterogeneous response. It's my guess.
Oh, thank you. Here's the last question for you, Dr. Belcher. So this question is related to what you mentioned about the hemopexin therapy. Do you think that there can be a detrimental effect when all this free heme is sent into the cells and has to be metabolized by HO1?
Yeah, I think, you know, it's really interesting why we don't have quite enough HO1. It's a very delicate balance, but if you get heme there, you get a bigger response with the hemopexin heme than you do with heme alone, it seems. And that may be to the signaling pathway, LRP1 signaling pathway. We don't quite understand why sickle patients, if you add just a little bit more HO1, it's protective, and why sickle patients don't already have enough HO1 to protect. I just think it's like a balance on a teeter-totter, you know, the kids play on. I just think that just a little bit of pro-inflammatory or anti-inflammatory can tip it either way, the balance in sickle patients. I think that balance is very delicate.
Okay, thank you. So okay, another question about glutamine or heme modulates IL-18 that might be responsible for the cardiovascular disease?
Boy, that's a great question. Also, yeah, I saw that paper. It's a very fascinating IL-18. Turns out that TLR4 activates signaling will also activate IL-18. And I think that's actually in the Boza paper, that 2007 paper I talked about. I think they also measured IL-18 production by macrophages. So it kind of fits with the, you know, with this TLR4 signaling and this IL-18 causing the myelofibrosis in the heart.
Okay, thank you. I think that's all we have in the chat. So if you have any other questions, please let us know and we can pass your questions to the speakers and they could follow up with you. Yeah, so thank you for our speakers' fantastic talks.
Thank you.
Thank you very much to both of our speakers. I want to thank you on behalf of the Society and the journal, and the webinar will be available on the SCBM site afterwards, won't it, Jessica?
Correct, yes. After processing, we'll post this online at scbm.org, so you can view this webinar in its entirety, as well as our previous two sickle cell disease webinars at scbm.org later on this week.
Okay, so thank you very much.
If you're interested in joining at scbm.org, everybody take care.
Thank you.
Thank you.
Bye-bye.